Good afternoon, everyone. My name is Franz, and I would like to present my idea, Ancient Marine Metabarcoding, a new approach to stomach and microbiome analysis for historical fish. And with historical fish, I mean museum fish. So, first of all, we have to start with museum collections. And there are plenty of fish in the sea, but there are also quite a few stuck away in natural history museums. Basically, shelves and shelves of fish in formaldehyde. And why are we not using them for genetics? Because it does sound like a haven for population geneticists. But most of them have been stored in formalin or formaldehyde, and that causes huge problems. First of all, it creates crosslinks between the DNA and proteins. It hydrolyzes the DNA, and furthermore, also fragmentation occurs based on age and bad preservation, for example, bad buffering. So my research question was, can we harvest some of this potential? And what do we want to do? We wanted to have a look at prey items and the microbiome composition in Antarctic fish. And the method we wanted to use was metabarcoding. So metabarcoding is basically a genetic approach of barcoding to identify certain species. And metabarcoding just means we're not doing it on an individual level, but we can do it on the whole stomach simultaneously and then find the diversity. The organism we wanted to study was Trematobos, an Antarctic fish, a genus of an Antarctic fish, and we collected 200 samples from the Natural History Museum in London. And they're quite closely related. They have slightly different lifestyle. There are 12 species in the genus. So our question was, what do they eat, and what is the microbiome, and is there any relation between what they eat and what the microbiome is? I'm going to jump directly to the results, and I first have to say, with CO1, so basically prey item composition, we didn't get any results that showed anything except for contamination. We are not entirely sure why this is, but yeah, that's what we found. However, with 16S, we did find, so 16S we use for the microbial community. So here you can see a PCOA plot of the microbial diversity. And the yellow ones are the recent fish samples we sequenced, and the museum ones are the blue ones are the museum ones. And we implemented quite a lot of controls like blank extractions, human contamination, workbench contamination, to make sure we are not actually just sequencing contamination and find at the end it's different to fish, but yeah, at the end. So what do we find here? So in the left, up here, we find the contamination and the recent samples, and then we find quite a lot of museum samples that are distinctly different to the controls, except for one control up there, which we think was also contaminated. So, looks quite nice on the first glance. Then we had a deeper look at the samples and tried to find any biological relationship. And as I said, the prey item composition we couldn't figure out. However, we had a lot of metadata like size or age of the fish, and we actually tried to correlate the size of the fish to the microbiome composition. And we did see that fish along the PCOA2 axis got bigger. And we also extracted the PCOA coordinates and plotted it against the standard length and found the correlation was significant. So that gives us a lot of confidence in that it is actually biological sound what we found. So one thing I have to say is sample dropout. So we first pre-selected 400 specimens from museum samples, which from the catalogs, which we thought was interesting. Actually, only 200 of them were suitable because for the rest they were too small or the guts were removed. Then only 130, the application was successful. Then sequencing was successful for only 80. Then quality filtering, we ended up with 50, and at the end, after we removed contamination, we ended up with 27 samples. So that is quite a lot of work and quite a lot of money on a few samples. So the overall conclusion, of my study of this last year I've been spending on this, is crate item analysis yielded little to no results. Maybe they were not well preserved, maybe there was something else going on, we're not sure. We are also still looking into this. But microbiome data showed to be different to recent samples, which is quite expected. Microbiome seems to be dependent on the size of the fish, which I think is a really awesome result. And it is biological sound, because if fish get bigger, they change their diet, and hence their microbiome does change as well. So it's nice to find something biological to prove that it actually worked well. So the big question is, I guess, for everyone who thinks now, well, should we do this as well? It is expensive, so I spent 5,000 euros on all of this for, at the end of the day, 27 samples. So a huge sample dropout, quite expensive. But if you have a great research question, I think it's unique, like it's amazing, because you can compare old microbiome to a new microbiome and see, for example, the effects of climate change, fisheries, or whatever. And I think that's a really unique opportunity. So I leave you with that. No, I think questions afterwards. So find me or shoot me an email if you have any questions. Thank you.